All right, good evening, everyone. This is our Unit 3 review. Sorry I couldn't do this in person. Like I mentioned, I got a lot of stuff going on. I'm trying to get this out here late at night for you guys. Um, real quickly, so the exam is going to be due on Sunday at 1:30 p.m. So you get a little extra time to do it. That way you're not rushing late at night. If you need the extra day to study, extra time to study, and then wake up in the morning, nice, refresh, and take your exam, you can do that as well. But just take advantage of the extra time to get some last minute studying or go over some of the concepts. The big stuff I'm going to go over is basically the different types of defenses. Um, Most of the other stuff, the antibodies, um, let's see what else is on here, blood typing, the types of immunity like natural, passive, natural, active. Those typically, there are a couple questions here and there, but a bulk of your exam is actually the types of defenses. And if you don't know this, you're gonna do bad on the exam. If you know this and you don't remember some of the other stuff, you can get by with a pretty decent grade. So I want to make sure you understand these. And then I'll go over practice cases, how to work them. You are going to get at least one, maybe two practice cases on this exam. And they're very good because they can test your knowledge on all the different things we talked about for the immune system. And so I'm going to kind of show you how to work through them. I'll do a practice one here in the session. That's also part of your study guide. So if you haven't completed that, I'll be doing that one for you as well and kind of showing you how to do that as for the other two. Okay, so the first thing is first line of defense. That's everything that helps keep microbes out of our body to begin with. So that includes physical barriers such as skin, respiratory system, the eyes, GI tract, reproductive system, and also chemical barriers associated with those organs and those tissue types. I broke it down like this because it's going to be more relevant when I start doing the practice cases. But if you don't have something similar to this, I would recommend you breaking down skin physical barriers and skin chemical barriers, respiratory, physical, respiratory, chemical, etc. So you can kind of see, especially for the physical barriers, most of them are very similar. So GI tract, reproductive, respiratory, they all use mucous membranes and mucus. You got cilia and respiratory and reproductive. Eyes, you really just have blinking. That's probably not going to be on the test. If I was giving you a test, I probably would put eyes on there, but just throwing it on there because it is part of your study guide. And then the skin, basically just the epidermis and the dermis. Chemical layers, that's where you get a little bit more differentiation between how your body can protect itself. You have different things like the acidic pH on your skin. When you sweat, you leave behind salt. You also secrete oil. We all have, some of us have oily skin. I know I do. And while it's very nice to get that off, it actually provides a very good line of protection to prevent microbes from actually growing on my skin. You can see respiratory system, lysozymes, lactoferrins, basically help prevent the growth of organisms, microbes and bacteria, helps destroy the membrane, that kind of thing. The GI tract has a lot of different things. If you don't really understand how your system works, mucus, the way it does, it'll ball up a bunch of microbes and it kind of directs it towards your stomach. So it wants you to swallow it. That's why you have so much phlegm when you're sick. And ideally, you can get that into your stomach where the hydrochloric acid can digest it. It's very acidic. And then if you've taken BIO202, you'll know that bile is more alkaline. And so anything that's acidic that can survive an acidic environment will then be thrown into a very alkaline environment in the small intestine and it'll break down there. So our body is really well designed to handle a multitude of different types of microbes. Reproductive system, this does apply for males, but primarily females because their openings are a little bit bigger. And so that opening allows for different types of microbes to get themselves in there. And you also have layers of protection there. Another thing to remember with first line is your resident microbes. I threw this image from here. It's in your handout, but you definitely want to know why the resident microbes help protect you. Basically, they're competing for resources. And if we're giving our resources to our resident microbes, there's less for invading microbes to eat and consume. Baby milk, actually the third largest concentration in baby milk is a certain material that we humans don't digest, but our gut bacteria does. And so getting baby milk, breast milk, helps actually form that initial gut biome. Just a little fun fact, not part of this lesson, but I just learned that a few days ago, so I thought that was very interesting. But ideally, you want to be able to know what types of microbes exist in different parts of the body. We've been talking about a lot of these already. Staph epidermidis on our skin, Staph aureus in our nose. For the vagina, specifically if you get something else with the vagina, you're talking lactobacillus and Candida albicans, for example. And then we know in our GI tract, we can have E. coli as well. Okay, second line of defense. So if those microbes make it through our skin, through our openings in our body and start getting into our deep tissues or into our tissue themselves, we have different chemicals or different cells that can help protect us. So I made a list of the cells, the macrophages, neutrophils. Understand those are involved in every immune response. And they primarily do phagocytosis, and then macrophages do a lot of other stuff like complement proteins, fever, inflammation, and they also present the foreign antigen to our CD4 plus helper T cells to start the third line of defense. And then basophils / mast cells, those are all allergic responses. Eosinophils are parasitic, so those are very specific to those types of infections. Again, this is in your handout. This is page 14 of your handout. I would recommend, if you still struggle with this one, I don't know if Dr. Carter brought it up, you're going to get a note card, one-sided note card. This is probably something very good to throw on that note card. It has a lot of information, and multiple different questions can be asked on this information alone. Okay, now we get to the third line of defense. The way I learned this is where's the infection occurring, and depending on where the infection's occurring is whether I'll have a B cell or a T cell involved. So kind of like I wrote here, so if the infection is inside of our body, you're going to have an intracellular response, or if you remember inside of your cells, and I said body, I meant cells. So inside your cells, understand that you are priority number 1 in your cells. So you're going to have a Th1 response. So macrophages are going to report to the Th1 CD4 helper T cells, and they're going to report using the CD4 plus receptor. After that, the CD4 plus helper T cells will then activate the cytotoxic T cells and memory T cells. Those cells are going to go through and they're going to check all of our other cells using T cell receptors to see which cells are infected and which cells are okay, kind of like the TSA checking your passport. A little bit extra information on this. The way this happens is your cells have that MHC1, the major histocompatibility complex. So MHC1 is your cell telling the rest of the body, hey, I'm you and I'm okay. The MHC1 can also tell the body, hey, I'm infected. You need to come get me and come get rid of me. So the CD8 cytotoxic T cells are basically checking the MHC1 and they're saying, okay, are you okay? Yep, you screen good. You look like you're supposed to be here. You're healthy. I'll move on to the next one. And then if it sees the opposite flag, then it knows, oh, you're infected. I need to get rid of you. It'll blast them with perforins and granzymes, make a bunch of holes in the cells. The cell will kind of rupture, and all those viral components, the proteins, maybe the viral RNA or DNA, that all gets outside of the cell into like our tissues, and that allows our macrophages to then come and start eating it all up and get rid of the virus. Unfortunately, to get rid of the virus, you do have to kill your own cell, but that's just how our body's designed. If the infection is outside of the cell, basically not a virus, it's a bacteria, parasite, an allergy, those reactions typically happen outside of our cells. And so those are priority number 2. So those are going to be a Th2 CD4 plus helper T cell. So again, the macrophage presents this to the Th2 CD4 plus helper T cell using the CD4 plus receptor. This will then activate the plasma B cells and the memory B cells. And the memory B cells will then look for that invading microbe using the MHC2 complex. I'm not going to go too far into that one. Oh, my bad. MHC2 was involved in macrophages. Forget I said that. But anyway, the B cells are going to start looking for the invading microbes. And when they find them, they're going to release antibodies. The antibodies don't kill the microbe. What they'll do is they'll kind of start attaching to the microbe, let's say the bacteria, for example. And this attachment allows, let's say I got attached to one bacteria here. I can use antibodies to attach to another bacteria, and I can connect them together and make this chain of bacteria and antibodies. And these antibodies, what they kind of do is they kind of clump them all together, and then they basically season the microbes so that your macrophages come in and start eating those up. So again, antibodies do not destroy. Instead, what they do is they agglutinate, which is the clumping part. They neutralize. They connect to where the bacteria would connect to other parts of our body. So it kind of blocks them from doing that connection. Opsonizing is the seasoning, making them nice and sweet and tasty. And then they'll also trigger complement fixation so that the other complement proteins can kind of come in and do what they need to do. I threw in here just to know your antibody types, know which ones are always involved. So you're always going to have IgM and IgG, but you might also have some very specific ones like IgA, part of the respiratory system, or IgE involved in allergic responses, and IgG typically takes over, IgE will typically be produced instead of IgG during allergic reactions. Okay, so next thing is how to tackle a case study. So again, on the exam, you're going to have one, maybe two case studies. And the way the case studies are going to work is you're going to be given basically a prompt, maybe a paragraph or a couple sentences explaining, let's say Luis was walking around and he tripped, bumped, scraped his skin, and he got Staph aureus into his skin. What are you going to do? You know, how would you treat it? How would my immune system respond to this? I put an example here. So step one, where's your infection and what is causing it? So I gave you this big blurb. I fell over, I was drinking coffee. None of that really matters. What matters is, what was the infection? Was it bacterial, parasite, a virus? And where is it? Where in the body is it located? The example I'm putting here is a bacterial infection into the vagina. It's very specific, very broad, but you might get a bigger prompt. Just know that a lot of that fluff doesn't matter. Look for what is it, what is the infection, and again, where is the infection? So step two, you're going to list your first line chemical and physical barriers involved in the response. So for the vagina, again, mucous membranes, mucus, and cilia. For physical, chemical, mucus, acidic pH, and lactoferrin. That kind of goes back to what I was showing you all earlier here. And that's kind of why I broke it down in this way. It's very easy for me to study this. Also probably very easy to fit on a note card if that's what you're going to do. And you can kind of see skin, physical, chemical, respiratory, physical, chemical, and I can just kind of go and work my way through that way. It groups them better for the case studies, in my opinion. Okay, so third step, are there any residents involved in that area? So if I had said, hey, I've got a cut, a bacteria on my skin, all right, I've got Staph epidermidis that can be there to help me out. In the vagina, I simply picked out Candida albicans and Lactobacillus acidophilus. Again, these are very specific. Kind of goes back to this page here. Just know a few resident microbes. You don't need to know all of them. Just the basic ones you should know: Staph aureus, Staph epidermidis. I would know your vagina ones and maybe your urethra one as well, because those we don't typically deal with. Or anything in the intestine, E. coli. If you know at least one, maybe two, you should be good. But one for sure. I'd know two just in case, just to be safe. Okay, step 4, you're gonna list your second line chemicals, cells, and processes. So the way I broke it down is I really talked more about the macrophages, but I understand the neutrophils also do their own processes, but not as many as the macrophages, which is why I want to list them here. So in any infection, second line cells that are always involved are macrophages and neutrophils. So those should always be listed, no matter what the infection is, where it is, you know, who it happened to, doesn't matter. It's going to be the same two cells. Make sure those are always listed. For the chemicals, I wrote digestive enzymes, interleukin 1, interleukin 6, and then your complement proteins, C3b, C3a, and 5a, and C5 through C9. And those combined, all those chemicals trigger some sort of phagocytosis, fever, inflammation, and the MAC attack. And then step 5, this is really where I think students struggle, and where I see your kind of exam scores start differing, is did you make the right response? Did you pick, is the infection inside the cell or outside the cell? Because that's going to determine whether you go with the Th1 or Th2 response, which then determines whether you're using B cells or T cells, which then determines if you're using antibodies or perforin gray enzymes. And you kind of see if you make the wrong decision on question 4, and you're still answering the next few questions thinking you're in an extracellular response, you're going to get all of those wrong. So whenever you get to this part of the question, make sure you understand, is the infection inside of my cell or outside of my cell? I.e., is it a viral infection or is it something else? Okay, so before I get to that good luck part, I'm going to go to the practice case. Maybe I'll make it a little bigger. There we go. Okay. I'm just going to show you how this could be presented in an exam. Again, this is page 9 of your study guide. So if you've already done these, great. You're basically checking your answer. If you haven't done them, awesome. I'm helping you do at least the first one here, and I'll hopefully make it easier for you to do the next two. So we're going to, again, start with the step 1, which is where is your infection and what is causing it? All right. I have a respiratory infection. That's my where. And what's causing it? The influenza virus. Pretty straightforward. So I'm going to start with my first line physical barriers. So again, if I go from here, my physical barriers involve the respiratory system. You're going to have mucous membrane. You're going to have mucus. And you're going to have cilia, or little nose hairs. For your first line chemicals, you're going to be looking at the lactoferrins and lysozymes. Resident microbes, again, I mentioned if it's in your nose, the easiest one is Staph aureus. I cannot spell today. Another one you could use if you're thinking about adding a second one is Staph epidermidis. If I spelled that wrong, no, let's say I spelled it right. Epidermidis. Okay, so non-specific cells, again, it's always the same two cells, macrophages and neutrophils. For your chemicals involved, so you're going to have, sorry, my dog's over there. She's just staring at me, waiting for me to be done with this. Digestive enzymes. I cannot spell. There we go. Fever. Nope. IL-1, IL-6, probably C5b, C3a, and 5a, and C5 through C9. So digestive enzymes help you with phagocytosis. Interleukin 1 is fever. Interleukin 6 is inflammation. CB5, C5b also helps you with phagocytosis. C3a and 5a is inflammation. And C5 to C9 is the MAC attack. And again, for third line response, this is where everyone kind of differs. So if I have an influenza infection, the location doesn't really matter in this case. Really, it's the type of infection. Is it a virus or is it a bacteria, parasite, allergy, etc.? So because I have a virus, that means it's going to be an intracellular attack, which means I'm going to need to prioritize myself. That makes a Th1 response. And another thing I'm going to throw in here, which isn't necessarily listed, but this is going to be a question on your exam, so you're getting a little bit of an extra heads up, if you will, is you're going to be asked certain questions about what receptors are involved in the response. So for this portion of the response, you're always going to have CD4 plus and T cell receptors, TCR receptors, but you know what I mean. The reason I want to throw this in here because I want you to understand when the macrophage is communicating with the CD4 plus cell, it's still communicating via a CD4 plus receptor. It doesn't matter whether it's extracellular or intracellular. Just know that if a T cell is involved, specifically the helper T cells are helper T cells, you're going to have a TCR, a T cell receptor, and you're also going to have a CD4 plus receptor because the cell is a CD4 plus helper T cell. I'm emphasizing it because you're going to be asked questions on what receptors are involved. You should definitely understand which receptors are involved in this step of the process, which is where the macrophage communicates with the helper T cell. Okay, next, that is going to trigger a response by the cytotoxic T cells. And the memory T cells. B cells, again, it's a Th1 response. There are no B cells involved. Because there's no B cells, there are no antibodies involved. And I kind of wrote this in here already, forgot to delete it. But if you're looking at cytotoxic T cells, those are out there to hunt and kill our infected cells. They're going to use perforins and granzymes that help make holes in the membrane and help digest the viral parts that are found outside of the cell after we pop our cell open. And that's it. So hopefully you're checking your answers if you've already done this and this looks good with your notes. If there's any changes, go ahead and make them now. Again, this is just practice. The study guide's just practice. Ideally, you're prepared for the exam. I would follow the same process to do these other two if you haven't done so. If you have questions on these two, just reach out to me. I can review those for you before the exam. Again, these are just there for you to practice. You are going to get a practice case on your exam. Guaranteed one, maybe two. You can kind of see how this one case, I could ask 1, 2, 3, 4, 5, 6, 7, 8 questions. And really, if you look at the second line, non-specific chemicals, I could ask multiple questions here. So you can kind of imagine on a test that's 80 points, this one question could be worth 10 to 12 points easily. And that's the difference between an A and a B. And so definitely you want to know these specifically, how to be able to work your way through the cases. The other stuff, if you forget, different types of immunities, it might be 1 or 2 questions. You're probably going to be fine on the exam. They'll study those. I'm not telling you not to study those. But if you have to triage what you're studying, you should be triaging first, second, third line responses, how to deal with the case studies, and then work your way down with all the other information. Okay, and that ends it for me. Last thing, I'm going to leave you here with a little joke. What is funny about humoral immunity? It's because plasma cells are involved with humoral immunity. I probably butchered that. Actually, I did butcher that. Just read the picture. It was funnier when I looked at it than when I'm actually trying to read it. But the biggest thing is good luck on your exam. Again, you have an extra half day to complete it. So it's not due at midnight on Saturday. It's now due at 1:30 p.m. on Sunday. Use the extra time to study. You can ask questions. If you're in labs with me tomorrow, you can ask me some questions then. If you're in the Monday group, again, feel free to message me. But definitely do the same things you did last time. Find a good place to take the exam, quiet environment. For the note card, I believe Dr. Cotter is going to hand those out if she hasn't already handed them out to you. And at the beginning of the test, you're going to show your note card to the camera, and then you're going to start the test. And that just lets us know that you're not writing anything else down, and that kind of thing. So again, reach out if you have any questions. Good luck, and I will talk to you soon.